Hi guys, my name is Online Med Nerd, and today we'll talk about the Malate shuttle as well as the Glycerol Phosphate Shuttle. So, what does the Malate shuttle do? It basically shuttles molecules between the cytosol and the mitochondria. What are those molecules? Malate, NADH, oxaloacetate. Malate is a molecule that can cross the mitochondrial membrane, so does not need any sort of transporter, whereas NADH and oxaloacetate cannot cross and therefore need a transporter. NADH is basically required within the mitochondria to take part in the electron transport chain in oxidative phosphorylation for the generation of energy in the form of ADP, whereas we need oxaloacetate in the cytoplasm to take part in gluconeogenesis for the generation of glucose for maintaining the blood glucose levels. So those are the basic two key uses of this shuttle. Now, if you focus on the diagram and follow the numerals in purple, and I'll read along. OAA stands for oxaloacetate, which combines with NADH, giving its electrons and forming malate and liberating NAD. Malate can therefore go through the protein transporter into the mitochondria, where it still contains the electrons, which now it transfers to NAD, forming NADH. NADH inside the mitochondria can now participate in the electron transport chain, whereas when it was outside the mitochondria, it couldn't because it wasn't able to cross the mitochondrial membrane by itself. Therefore, it needed this transporter. NAD now is gone to the electron transport chain. Oxaloacetate, which was liberated, now combines with glutamate, giving off alpha-ketoglutarate. Alpha-ketoglutarate itself can cross the mitochondrial membrane back into the cytoplasm, whereas aspartate cannot, so it goes through a protein transporter, as seen in this figure. And then it can regenerate oxaloacetate in the cytoplasm, as seen. Now we're flipping over to the other side. In the cytosol, the second function was to transport oxaloacetate from the mitochondria into the cytosol. So, following the numerals, oxaloacetate OAA combines with NADH, forming NAD, giving off its electrons to and the formation of malate. Malate can now cross the membrane through the protein transporter into the cytosol, where malate gives its electrons back to NAD to form NADH and liberating oxaloacetate. Oxaloacetate can now take part in gluconeogenesis for the generation of glucose for maintaining the blood glucose levels within the body. Now, our second shuttle was the Glycerol Phosphate Shuttle. In the Glycerol Phosphate Shuttle, as mentioned in the numerals, follow the 1-1, then the 2-2, then the 3, and the double 4s, and 5. So the first is dihydroxyacetone combines with NADH. When we have glycerol phosphate dehydrogenase enzyme catalyzing this reaction, liberating NAD and giving off glycerol phosphate. Glycerol phosphate, still on the cytosolic side, now undergoes a reaction using the mitochondrial membrane glycerol phosphate dehydrogenase and FAD on the inside of the mitochondria, giving off FADH2 and giving off dihydroxyacetone on the cytosolic side. So basically, with these shuttles, we've been able to generate NAD inside the mitochondria or FADH inside the mitochondria, and depending on the type of molecule we've generated, the electron transport chain can take these and generate ATP in different forms. If we have a molecule of NADH, we will get 3 ADPs, and if we have a molecule of FADH2, we will get 2 ADPs. Thank you.